Hi, welcome. Thank you for checking out my poster. This is Harjeet Kara from the Lee Lab. Today I'm going to be talking about my project, which focuses on characterizing genetic mechanism involved in measuring day length, also called photoapeirism, in Drosophila melanogaster. Photoapeirism is the organism's ability to interpret changes in day length. It can influence survival and reproduction of many organisms. It can also impact human health. For example, it has been studied that during spring and summer months, when there are longer days, there's a rise in manic episodes. Then during fall and winter months, when the days are shorter, there is a rise in depressive episodes. Another timekeeping mechanism that is present in the organisms is called the circadian. The circadian clock is a regulator of many metabolic processes and derives a 24-hour circadian rhythm. However, it's not well understood how the circadian clock plays a role in photoapeirism. In this project, we aim to better understand the genetic mechanisms of photoapeirism using the Drosophila genetic reference panel. We have hypothesized that by performing a genome-wide association study, we will be able to identify candidate genes involved in photoapeic regulations. We also aim to understand how the circadian clock and photoapeirism interact to interpret day length changes. We have hypothesized possible interactions between the clock and photoapeirism. The first possibility is that the clock and photoapeirism do not interact at all. The second possibility is that either the clock controls the responses of photoapeirism or photoapeirism regulate the responses of the clock. The final possibility, and the most likely possibility, is that there is an overlap in mechanisms between the circadian clock and photoapeirism. In the present work, we measure the well-known phenotype that is under photoapeic regulation, called the cell coma recovery time. CCR is the time it takes for the flies to recover from a paralysis-like state after being kept under cold temperatures. In this study, we used two different photopairs, long day and short day, and we induced the chill coma by placing the flies in 4 degrees for 60 minutes, and recorded their recovery time by the Drosophila activity monitor system. We found that there is a variation in recovery time among the DGRP genotypes. In Figure 1a, two representative DGRP genotypes are shown, KLS57 and KLS69, under different photoapeic conditions, which have different recovery time from each other. In Figure 1b, we calculated the Cohen's d because our primary interest is to measure the differential response of each genotype to two different photoapeic conditions. Cohen's d can be divided into different effect sizes, large, medium, and small. After performing the GWAS analysis, we performed the gene enrichment analysis of the top GWAS hits. We found that certain genes are enriched as shown in Figure 2a. From the top 40 hits of the GWAS, we narrowed down the photoapeic candidate genes to top 40 hits by their significant p-values, shown in Figure 2b. Next, we looked at the photoperiod response of the knockout mutants of the candidate genes compared to the wild type in Figure 3. Since we were interested in knowing the difference in photoperiod response of the candidate genes from the wild type in Figure 3b, we calculated genetic effect size. It was found that 3 candidate genes show a significantly differential response from the wild type, with PKC delta being the most different. We also tested a period of these knockout candidate genes to test the hypothesis whether these photoperiod candidate genes would be involved in circadian regulations. It was found that GLC-ATP had a significantly longer period compared to the wild type. We also wanted to test the well-studied dye-pause response of photoapeirism to confirm the CCR assay results. We chose to look at the dye-pause response of the PKC delta because it showed the most difference in photoperiod response from the wild type. We tested 2 mutants of PKC delta and found that P-back mutant and wild-type flies showed significant difference in dye-pause response between short day and long day, but not the MIC mutant. Next, we tested the hypothesis whether well-known circadian clock genes would be involved in photoperiod response. After performing a CCR assay, we found that compared to the wild type, the clock genes showed a photoperiod specific response. The genetic effect size showed, in compared to the wild type, period short showed a significantly differential response. In conclusion, by performing a GWAS analysis, we were able to determine the photoperiod candidate genes. Our results also showed that one of these candidate genes, GLC-ATP, might play a role in circadian clock regulations. The circadian clock genes also showed a photopair-specific response, indicating the hypothesis that there are shared regulations between the clock and photopairism.